you mentioned Michabo Health Science and the university. The reason for having both affiliations in this talk is I'm drawing on material from both affiliations, so or both organizations. So that's why I've presented both on the bio and on the introductory slide. And to provide a bit of rationale behind those of you that do know me and know me as almost a lifetime academic, what we discovered a few years ago was we really wanted to push the NAMs and, in our case, the omics out towards genuine applied scenarios and applied case studies. Universities are not necessarily, or perhaps UK universities are not necessarily the best at interacting with businesses and regulators, and so we actually discovered that by pushing out a commercial activity, it actually makes working, it's basically extended our ability to work with companies and work with regulators far more easily. So it's broadened the portfolio, and it's been a really good and exciting move. We did that in 2018. So that's the reason for two institutions listed. Okay. So in today's keynote, well, first of all, not only thanks to Carol for the introduction, Connie, thank you for the invitation, and to the committee. It's a great honor, and hopefully I can update you on where metabolomics has reached and show you some highlights over the next 35 or 40 minutes. So in terms of today's talk, I want us to just step back for the first few slides. I don't want to just jump in. In a sense, whenever you go to workshops, conferences, and if you're talking about metabolomics, there's always there's a sort of a question that lurks. Don't we have enough omics? Doesn't transcriptomics already give us enough information? And so I want to start the talk and introduce, just show you a few slides and make a case for why metabolomics should be part of the conversation around NAMs and the regulatory applications of NAMs. I will also admit immediately that in almost every one of the tox studies that I'm involved in, whether it be in the university or in Michabo Health Science, we use transcriptomics as well as metabolomics. So I'm a complete believer in transcriptomics, and I'm not somebody who's, I'm not technology pushing on one particular technology. Quite the opposite. See the value in bringing together different omics. And I inserted one slide to help draw that point out. So I want to just show you a few introductory slides on metabolomics in case some of you are less familiar with it, and position the work a little bit, and really ask the question, is it relevant? Then get into this case study. We have a number of multi-omics case studies in chemical grouping. And we're very close to being able to release publicly 2 more case studies, but we're not there yet. And so I'm going to show a case study in azo dyes that I know a number of you will have already heard me talk about. So bear with me on that. I will present that, draw out some of the, we made progress, but we really identified some of the challenges. And it's the challenges that were identified that I go on and talk about more in the following slides. In the third bullet point here, I will introduce to you a project that was funded by the European Chemicals Agency and has led to the development of a metabolic biomarker list. And I'll say more about why we did that and a little bit on how we did that in the middle of the talk. And we were doing that ultimately to strengthen the mechanistic interpretation of our omics-based grouping studies. So you'll see in gray, I've inserted grouping in various places through the talk, because from our particular focus is on pushing this omics-based grouping, but actually a number of the topics I'm going to be introducing here actually have broader application, broader relevance than just grouping. Just a few slides, 3 slides on what I've listed here as an inadvertent application of metabolomics. And it's really an underexploited benefit that it was about 3 or so years ago that we turned around something that used to be a problem for us, and we turned it into something that's actually a benefit to us. And so I'll say more about this inadvertent application of metabolomics to actually provide information that relates to metabolism and even kinetics than to biological response. So this is actually raising quite an interesting prospect for being able to get toxicodynamic and toxicokinetic data out of the same samples and the same assays at the same time. So I'll just touch on that, just 3 slides. And then partly because I knew Carol was introducing me, but it's actually highly relevant to talk about it. Just in the last 3 slides, I'm going to touch on just a snapshot of progress on where we've reached on 2 other very important topics. What is the reliability of these approaches, reliability of metabolomics, and more broadly, but also in terms of grouping? And how are we doing in terms of progress around the reporting of omics data and actually specifically the reporting of omics data, again, for grouping. So some of my highlighting here, the relevance of the assay, the reliability of the assay, reporting of the assay, and ultimately validation of these assays. These are some of the key words that we, of course, know we need to be considering. Okay, so why introduce metabolomics into this current very interesting field of new approach methodologies? So reason number 1, you're gaining information on mode of action. And if you gain information on mode of action, you're working towards hazard identification. And okay, so you could say, well, any of the omics provides information on mode of action, and this is absolutely true. So why would we argue to introduce the metabolomics? And it's because in the metabolomics, you are measuring the most downstream of the molecular responses. It's the most downstream ohm. And hence, by being downstream, it's closer to the phenotype of the biological system that you're studying. And those in metabolomics have been aware of this for a long time, and I decided to dig out, and this is a paper from 12 years ago, my group, when we first started trying to evidence that through studies of where we're looking at an adverse phenotype. This was actually looking at energetic status as a phenotype, and then we're discovering metabolic biomarkers that could actually predict that phenotype. So published in Toxicology, as I said, 12 years ago. Somewhat more recently, using metabolomics, again discovering metabolic biomarkers that are predicting chronic reproductive toxicity. Both of these applications are ecotox, by the way. But the point is, metabolomics definitely has the capability in the metabolic biomarkers of predicting aspects of mode of action, perhaps even adversity. Second topic, and it is interesting to see, of course, you have one of your focuses within this committee on points of departure, molecular points of departure. Just as you can apply BMD modeling to transcriptomics data, you can apply it to metabolomics data, and a number of publications exist. And so there's that benefit. And then the third, I've already just introduced on the previous slide, and I want to revisit today, this idea of not only extracting toxicodynamic information from a metabolomics assay, but actually obtaining information on the parent substance and its biotransformation products. So definitely overlap with transcriptomics, but it's the downstream phenotype that we're measuring that would be the justification for introducing metabolomics. This is the reason why we, in the work we tend to do, couples transcriptomics and metabolomics, because we're getting upstream and downstream molecular key event information. Certainly, application of both omics. Of course, you have to choose your gene or your pathway, or somehow derive a point of departure. Different ways of doing that, of course. And again, I would argue the metabolic point of departure may be different from a transcriptional point of departure and may carry different information. And then this third point is unique to the metabolomics assay. Now, am I talking about some sort of fundamental transformation? I'm waving this flag saying, let's bring metabolomics to bear in regulatory toxicology. Well, I'm a bit late to be the pioneer because it's been done for a whole number of years. So I'm going to evidence that with simply a few snapshots from probably one of the most used OECD test guidelines in the world, 90-day repeated dose in rodents. And what are some of the endpoints, molecular endpoints measured? T3, T4, estradiol, testosterone. I mean, these are all metabolic biomarkers. The way in which the assay is applied, it's a targeted assay to measure specific metabolic biomarkers to provide information, in this case, on different endocrine disruption. So there's nothing new about using metabolic biomarkers to inform on mode of action. And the next slide just sort of gives a few more examples of that. And a little bit of history, actually. I picked this one. This isn't toxicology, this is disease diagnosis, but it's, we all like to think we're doing state-of-the-art science. It was actually an English physician back in 1679 who first realized that you can diagnose a diabetic patient. They didn't have mass spectrometers in 1679, of course, so this physician used to taste the urine, and if it had this honey taste, that was the state of the art in the diagnosis. But using metabolic biomarkers for the diagnosis of a disease over almost 350 years ago. If some of you are aware of Stemina Biomarker Discovery, the US-based company, they have a couple of kits. Their developmental tox kit, and they also have a cardiotox kit. They're measuring small molecule biomarkers as well. So in the developmental tox assay, they measure ornithine and cysteine, and they take the ratio, and that's the predictor of developmental tox. So there's nothing that new about the concept of using metabolic biomarkers for informing on different modes of action. And I thought I'd round out this brief introduction by showing this particular image, which is lifted from the OECD's website on omics, just to make you all aware that the OECD, as Carol was mentioning, is certainly advancing the use of omics in regulatory tox testing as well. Now, the other reason for showing this image, because we have our favorite omics up here, and so in the case study I'm going to talk to you about, it's actually using both transcriptomics and metabolomics. And then we have, you could say, the low-hanging fruit for the applications of the omics. And with grouping read across here, that's a very nice way of leading right into this case study that I want to present. So this work, from my perspective, is work done in Michabo Health Science, and Michabo is a contractor to the European Chemicals Agency. So that's the reason why these two organizations have undertaken this particular study. And acknowledge my colleagues here at ECHA. A little bit of background, I'm not sure on the diversity of the audience, so I'll just say a few words on grouping and read across. Certainly in Europe, it has been an important alternative test method for a number of years, but it seems that grouping approaches, not necessarily grouping and read across, but grouping approaches have gained momentum considerably in the last 2 years within ECHA's internal workings and internal risk management strategies. So it's the most commonly used alternative non-animal test method in Europe. It sits formally within REACH Annex 11. And so the concept of forming a group of substances based upon similarity and then reading across or predicting tox data from a data-rich substance across to a data-poor substance. And of course, the key challenge is how do we actually assess the similarity of the chemicals to know that we can reliably put them into a group of similar substances? And conventional approaches for doing this include structural similarity, looking at phys-chem properties, looking at metabolism and looking for shared metabolic products, and using ecotox or tox properties. So various ways of trying to reliably group a series of substances to read across tox data from across those substances. And clearly the omics would fit here. So we're looking at introducing omics measurements to provide measures of toxicological similarity to strengthen read across, to strengthen the grouping hypothesis to enable more reliable read across. Now, why did we get here? Why was this a project that ECHA was very keen on undertaking? And that's because while ECHA's really strongly advocated using grouping read across for a whole number of years, on the right-hand side, I show the front page of their RAF, the read across assessment framework, which is not so much a guidance document, but a description of how they interpret grouping and read across. And it's a form of guidance providing information on the sort of data that should be submitted. And so they are, and have been for some time, very strongly supportive of grouping and read across. Yet, if you look at the percentage rejection rate, it's very high. The rejection rate of dossiers that industry has submitted to ECHA under Annex 11 using grouping read across, ECHA's rejected the majority of the read across arguments. And they publicize why. They have a report every 3 years, and so there's some quite well-established reasons for the rejections, and one of which is this scientific rigor in defining the group. So they are keen to find new approach methodologies that strengthen that grouping hypothesis. So the basis to this case study is, can we increase the acceptance rate of grouping read across dossiers, ultimately by strengthening the grouping hypothesis using what we sometimes refer to as molecular mechanistic data or omics data. So we undertook this case study and just run down through the steps that we conducted. So working with ECHA, first thing was we needed to choose a group of substances. And so this is the azo dyes, and I'll explain to you on the next slide, or introduce those dyes on the next slide. Secondly, we applied some conventional approaches to the grouping because we wanted effectively a baseline. This is what the structure or the conventional approaches would provide. Then we introduced the omics approaches, and we used transcriptomics and metabolomics to group chemicals. And I'll show you the results from that grouping. And maybe as a spoiler alert, there's a high degree of similarity, fortunately, between the conventional and the biological response or omics approaches. And once we had firmed up on our particular source substance, we then conducted the read across, and I'll show you that just very briefly. And there's one final part which I'll explain when I get to the slide. We actually mapped some of the omics data back onto structure, which was quite interesting. I'll show you that in a second. So let's go into the substances themselves. 7 azo dyes in total. The target substance is dispersed yellow 3, shown here. And we identified 6 possible source substances, shown on the right-hand side. A couple of Sudan dyes. So they're all azo dyes, but Sudan 1 and Sudan red G. A couple of dispersed orange dyes and 2 dispersed red dyes. And what we wanted to do was use the conventional and the omics approaches to work out amongst these 6 possible source substances, which one of them is the optimal source to read across from data rich on the right side to a data poor substance over here on the left-hand side. A couple more words of detail here. If you're interested in or know more about the RAF, this is a RAF scenario 2. So it's a very simple case of finding a single source that we have a single target, and it's a direct read across. So that's the scenario in which the case study was embedded. The test system actually was Waterfleet Daphne Magna, and the omics data was obtained during a standard 48-hour OECD test guide Daphne acute study, and then the endpoint that was read across was chronic, the 21-day chronic repro tox study. Now, what's the important point in red at the bottom? ECHA was not specifically interested in the azo dyes, nor were they specifically interested in demonstrating this or saying that this is solely relevant to Daphnia. Both the azo dyes and the Daphnia test system and the substances were, shall we say, convenient choices. The point of the study was to demonstrate how the process could occur, what it would look like, and what we can learn by undertaking such a study. So, 2 slides on conventional grouping. I'll do this quickly. The first conventional approach was using the QSAR toolbox. This was actually work that colleagues at ECHA conducted themselves, and using various QSAR profilers. The table shows you the outputs. Here are the 7 different dyes, and I'll go straight to the conclusion as you're looking at the slide and say, slightly unhelpfully, the target substance tends to sit on its own, of no surprise given this is based on structure. The 2 Sudan dyes are considered very similar and within a group, and the 4 dispersed orange and red dyes also sit together. Now, it's not quite as simple as there's 3 quite distinct groups here. Both the target and these two Sudan dyes, they're all phenolic, and this was flagged up in the structural profilers. So conclusion is DY3 is more similar to the Sudan dyes than it is to these 4 dyes at the bottom. And we then went on just to do a pure structure-based similarity assessment, and this is work we did in Michabo, very simple, and it's exactly the same message. So I'll show you quickly. This is structure using toxicology chemotypes as the fingerprint, and this is a dendrogram output showing the similarity between the structures. So we have a distance measure here. So the way to interpret these is, as I'm sure you know, where you have a branch point at a short distance, this is implying very high similarity here. So no surprise, 2 dispersed red dyes, which are structurally very similar. You would expect this. They have this node here at a low distance. Whereas our target, dispersed yellow 3, it's sitting up here. It's sitting closest to the 2 Sudan dyes, just as the information we got from the profilers. So as I said, this slide and the previous slide, very similar. So what we could take home from this at this point is, if you were forming a grouping hypothesis and you were working from the 6 possible source substances, you would be saying, well, it's one of the 2 Sudan dyes that we want to be using. Don't know which, but certainly a Sudan dye to read across to dispersed yellow 3. Now we go straight into the omics. So before going to the slide, I'll briefly say what we did. 7 dyes. Now each dye, exposures were conducted at low, medium, and high dose, times 7 dyes. So we have 21 different treatment groups. So what I'm going to show you on the next slide is a dendrogram, and it's a dendrogram based not on structural similarity, but on the multi-omics similarity. And there's 21 different treatment groups, so you're going to see a dendrogram with 21 branches. And so at first sight, it looks a bit, and it's rotated from the other side. So you've got distance here. And if we just go and look at the branching, say here, where you got this cluster of 3, which has a low distance score. So very high similarity here between whatever these 3 branches are. And if we read down, this is Sudan red, low, medium, and high doses. These 3 have a very similar multi-omics response. This is the metabolomics and the transcriptomics concatenated together. So let me highlight a few points. First of all, if you look across the bottom here, you will see the doses always group together for a given dye. So low, medium, and high dispersed red 13, and so on as you go across. And this is, in a sense, actually quite reassuring. And this is telling us that dye to dye to dye omics responses vary more than the relatively subtle differences between the omics responses within a dye across 3 different dose groups. These were factor of 3. So from low to high, that's 1 order of magnitude dose difference. But where it gets much more interesting is when we say, well, where are the groups? And in particular, where is our target? Because it's our target that we want to know where is the closest source to the target. Here's our target sitting over here on the far right. And look at this, the 2 Sudan dyes again in with the target. And through bootstrap resampling, we've got a 94% confidence based upon the omics data that this is a reliable grouping. We can push it a bit further than that and say, well, actually, which is the closest source substance to the target DY3? And we look up here, and with 89% confidence, it's Sudan 1. So based upon the omics data and the similarity of the omics responses, this is certainly a group with high confidence, but it's Sudan 1 that is the most similar to dispersed yellow 3. Now, a slide I don't typically show, but I thought I would today. As part of the analyses, we also stripped apart the transcriptomics data from the metabolomics data. And we said, well, what does each data type, what information does each data type provide in terms of this grouping? And what you will notice on the transcriptomics, and did it put DY3, the target, in the same group as Sudan 1? Well, yeah, it did here. That's all good. But look at the confidence score. And I'll flip back to the previous slide to remind you, 94% confidence in the well, actually, Sudan 1 and dispersed yellow 3, 89% confidence in that grouping in the multi-omics. And here, looking at these within this structure, 63% confidence. So confidence levels come down quite significantly. In a sense, it's even worse for the metabolomics data because we've got this strange medium dose DY3 has somehow, well, it's no longer lying within the rest of the Sudan 1 DY3. So we've got 72% confidence, but that's not really an accurate measure because one of the 6 treatment groups is not where we would have expected. So that's intriguing. And it's intriguing in the sense of, well, it would be logical that if you put together transcriptomics and metabolomics and different parts of the molecular response, to put those together, it is logical that you're building a stronger picture of the mode of action and better representing that in that fingerprint. And subsequently, you get a higher confidence in the grouping. And so I'll put that out there as a hypothesis. We don't have any evidence beyond that currently. Okay, so forming the groups through statistical grouping, that's okay, that's valuable, but that's not enough for the regulator. There needs to be some form of mechanistic interpretation. And I'm not going to go into this slide in any detail, but I will now, perhaps a little bit painfully for me, have to admit that this is where the transcriptomics is a number of years ahead of the metabolomics. So you have various options with the transcriptomics data analysis. And here's one example, just to put something on this slide, where you can get pretty reliable interpretation of pathway perturbations, at least pathway perturbations, using the information that you've measured in the transcriptomics. That is not necessarily the case with the metabolomics, and that was partly what justified the creation of the MTOX panel, which I'll tell you about in a few slides' time. So that's an absolutely obvious statement. If you want to look into what's driving that grouping and why Sudan 1 and DY3 are in the same group, or in this case, the 2 Sudan dyes and DY3, if you want to look into that and understand and try and come up with some mechanistic rationale, you of course need to know how individual genes, metabolites, how pathways, different gene pathways, metabolic pathways are actually associated with a mode of action. So I'm going to come back to that because that's not the case, hasn't been the case for the metabolomics. Just to finish the grouping read across very quickly. So what we identified is Sudan 1 as the source substance, data rich. Here's the Daphnia chronic tox data. We simply read across. So these are predicted values for dispersed yellow 3. Okay, the most basic of prediction, most basic of read across from a single source to single target. given that this is just a Daphnia 21-day test, we simply went into the lab and measured this. And so the measured 21-day tox data for dispersed yellow-3 is shown here, and here's the predicted above it. So hopefully you will agree that we picked a reliable source substance and confirm this through this experimental step. Okay, yeah, let's do this one quickly because I'm going to begin to not run out of time immediately, but I will run short on time. We were intrigued by the biological based grouping. So it's a dendrogram on the left. This is the omics-based grouping, and I'll just pull up this. This is our group of 2 Sudan dyes and the DY3. This is the omics-based grouping. And we said, well, what is it structurally that can put these 3 substances in one group and then the other dispersed red and orange dyes grouping separately from these 3. And if you look across the 729 structural fragments within the toxprint chemotype, there's only 2 that are unique structural features unique to this group, which do not occur in this group, and it's the hydroxy or the phenolic group. So, I mean, it's just a hypothesis, but based upon this analysis, we can hypothesize that if there's a structural feature that's driving this biological difference, biological grouping, that would be the structural feature that we would consider as a good candidate based upon this analysis. Okay, so where did this leave us? A few take-home messages. We demonstrated how the omics data can actually provide a quantitative measure of toxicological similarity, and it allowed us to group the substances, put a confidence measure on that, and importantly, to identify a single source that could be used for the read across. Conducted the read across, then went into the lab and actually confirmed that. And what's come out of this piece of work and other projects we're working on and many other good pieces of work that are published already in the literature, what we're working towards, and Eka's quite comfortable with this, is so far this sort of 3-step approach is, okay, you've got to design the study carefully. There will be a degree of using statistical methods to actually conduct the grouping, derive quantitative measures, but there really needs to be this more detailed inspection of what are the pathways that are being perturbed. Is your source and target chemicals really inducing similar pathways, ideally informing or providing some information on mode of action? It's something that we aspire to, can't necessarily claim that it's possible in every study. And in terms of this 3rd step, I've not presented any form of coherent argument to you for the case of the azo dyes because it doesn't exist. And it doesn't exist for a number of reasons, and I'm not going to go through all the reasons, but a really important reason is we couldn't interpret the metabolomics data in a manner that we wanted to interpret it because we didn't have the tools available to enable that form of interpretation. So we got a very limited ability to interpret that metabolomics data. There's analytical reasons as well. 4 years ago, the assays were quite different, and we're 4 years much further on now than 2018 when the data was generated. But the MTOX study was motivated in part by this particular limitation. Also questions around, well, okay, fine, we've got this case study, we're using omics data, how would we report that? How reliable is it? And we come back to those points that I'll touch on in my final few slides. So let me move into the MTOX. I've only got 3 slides on this. When you begin to interpret and look at metabolomics data, if any of you have experience in this, you will know pretty rapidly that there are few information sources that you can look at and obtain information on. For example, the linkages or associations between metabolic biomarkers and modes of action. And Eka funded piece of work which we published, co-authored with Eka in Feb, I think it was, of this year, published in Toxi, where we spent a year actually sourcing as much information as we could on metabolic biomarkers. And so we were looking at some pre-existing assays. There are some databases, such as the AOP Wiki, that does have some information on metabolic biomarkers. It's rather limited. Some other databases you'll recognize here. And it was actually digging into the primary literature that generated more than a third of the proposed metabolic biomarkers. And this was quite painful because we were manually examining close on 1,000 research papers to do this. So in essence, it was a big attempt to defragment, if that's a term. Well, it is for hard drives, so I guess I can use it. And we created what we call mTOX version 1, 722 metabolites that are associated with either an adversity or a mode of action, and pulled this together into a publicly available list. Version 2 is already underway. We've just got some additional funding for this. So we're going to carry on maintaining this open access list, this open access resource. And this list logically will grow as new biomarker discovery work is conducted. But we may lose some biomarkers from it, because if you've got, there may be some really unreliable markers in there, and over the next year, 2, 3, 4, 5, as more studies are done, it might be that certain markers just drop off the list. So at least we now have a place, a go-to point, a one-stop shop, I guess, for metabolic biomarker information. Yes, briefly, let me say a few words here. So these are some of the stats on the 722 markers. So 80% of those markers, for example, are associated with adverse outcomes. 64% are associated with pathways. We also, because you could perhaps see the analogy, if you know of and use the S1500 plus gene panel that NTP developed and published a few years ago, the mTOX 700 plus, you can see the analogy between the gene panel and the metabolite panel. Something we were quite interested in is looking at how the metabolites that we've got in the mTOX panel and the metabolic pathways, how do they relate to the genes in the gene pathways on the 1500 plus panel. And there's a 58% of the metabolites in mTOX lie in pathways that the S1500 plus panel is also interrogating. So good, there's a degree of overlap there, but there's also 301 metabolites which are accessing information which is not part of the pathways on the 1500 plus panel. So I think a nice complementarity, but I can see that the transcriptomics will make a unique contribution, as I believe the metabolomics can as well. Okay, and there's, on the right-hand side, there's some names, because I wanted to make the panel a little bit more alive, so put some metabolite names there, but not to dwell on that. Last slide that relates to mTOX. mTOX is a list of metabolites. It's a list for which studies in the past, or assays in the past, existing knowledge says these metabolites have relevance to predicting modes of action. It's not a biomarker panel that's implemented and you could sort of buy it off buy it as a kit or anything like that. However, state of the art these days in metabolomics assays, so the last year or so in particular, the assays are what we term hybrid. And hybrid means they actually have a combination of untargeted measurement and targeted measurement. And I'm definitely not getting into the analytical chemistry today. There are reasons for this, and the important part of this, and why I wanted to just show it on this slide, when we think about what should we target in a hybrid toxicometabolomics assay, well, that's now obvious. We target as much of mTOX 700 plus as possible, and that's in the Phenome Center that Carol introduced, which is our toxicometabolomics center in university. We're building mTOX into our metabolomics, into our hybrid assays, so that we can attempt to measure as many of these as possible. Okay, so 3 slides now, a little tangent, 3 slides on an underexplored benefit that the metabolomics assays can bring. So what I call the inadvertent application, and that is that arises because here's a wonderful mass spectrometer, thermo-scientific. This is a Q-Exactive Plus. Mass spectrometers in metabolomics, they are high resolution, high mass accuracy. They are mass spectrometers of a particular configuration that are really well placed to measure the test substance and the biotransformation products. And in fact, as I say here on the slide, if you take an biological sample from an exposed either cells or animal, and that sample is prepared and then analyzed by LC-MS metabolomics, you will be detecting, depending on the concentrations and so on, you really have the potential to detect the parent and possible BTPs, biotransformation products. And it opens up this possibility of thinking about how much information can we really get from one sample, from one assay. You know, you're measuring the endogenous response, but you're actually getting information on the some aspects of the ADME or TK. 2 quick examples, 2 slides. All this work has actually come out of projects working with what's now, I believe, called Division of Translational Toxicology. But I can't find a DTT logo, so I'm still using the NTP logo. There was a rodent study conducted several years ago for the purposes of transcriptomics that the NTP undertook, looking at triphenylphosphate, and I'll say it quickly, that we've been involved in a project with them looking at the metabolomics, looking at the endogenous biological changes and using metabolomics to probe those changes. But we said, well, what else is in the data set? What else have we inadvertently measured? And on the right-hand side, here's the parent, and here's a bunch of phase 1 and phase 2 metabolic products. All of those molecules, all of those biotransformation products and the parent were all detected in the metabolomics assay, inadvertently detected. And if you go back to a paper published in 2013 that studied the metabolism of this compound, all but one of the known biotransformation products was detected in our assays, and we picked up 3 additional products. So there's quite a lot of information in these samples. Second example of that, another study, this happens to be a phthalate, and you can see that fairly rich metabolism. All of those features were detected in our LC-MS-based metabolomics. Switch to something a little different, and that's all biotransformation products. What about the parent? Well, okay, you have to tweak your assay a little bit, but it's not difficult. We're now absolutely quantifying parent substances in the samples in which we're measuring the endogenous responses. So this is some very recent work looking in HEPA-RG, looking in both cell extract and media. I'm not going to go into the details here, there's no time. Happens to be a bisphenol. Got a few time points here, so we can do absolute quant on the parent. We've got evidence of internal dose, and you can do some basic kinetics on this as well, if you wished. So having to sort of say that very, very quickly, because I'm very conscious of running out of time in the next couple of minutes, I really think there's a lot of valuable information that historically metabolomics labs have been throwing away. And I think we really have to focus more on pulling this information out, because bottom line in a lot of the risk assessment work is cost. And if you're putting a sample into a mass spectrometer to measure the endogenous response, get information on the metabolism and the parent compounds at the same time. Final little bit, which I'll try and do in about 2 minutes. I wanted to give you it really would be a snapshot, and it clearly will be now, a snapshot on some of the other challenges that we're well aware of in omics and omics-based grouping. And this slide is on reliability, and the next slide is on reporting. So on the reliability, there is work underway. Carol already mentioned it, actually. There's a lab reproducibility study that's funded by Cefic that's underway. I so want to tell you the results from this, but we're still blinded. We still can't, we still effectively, it's embargoed. It may be about 4 weeks away before we can begin to reveal information, but certainly by the end date of the project, which is only 12 weeks away. So very soon we will be able to share information. So far, the results are really encouraging. These are the partners. It's an entirely blinded study, and ECHA has been very good at providing an independent assessor type role here. So there's that, which I'll share more of, love to share more about in the coming couple of months. And we've got a paper under review, a consortium of universities in the UK have a paper under review, which should be accepted in the next 7 or 10 days in Nature Protocols. And it's looking at accepting that we need some level of validation criteria. Of course we do for omics assays, for metabolomics assays, but it's introducing a tiered structure, 4 different tiers, depending on what context of use is. So can't really say anything more about it in any case until it's published, but it's based on the reviews, it's very close now. And that could be shared if anybody's interested. And yeah, sorry, Carol, I'm not going to do us justice here. In fact, by your introduction, you actually have introduced a couple of key projects. Reporting of omics data, you mentioned this. This is the HESI funded merit project. That was a forerunner to the OECD metabolomics reporting framework. The pleasure of working with Carol and also Josh Harrell, EPA, as the 3 of us have co-led what's become known as the OECD omics reporting framework, the OORF, which is very, very close to its final acceptance in the OECD review process. But 2 other points to note that are more specific to grouping, because this excellent piece of work has been more generic use of omics, not targeted on any one particular application. So the OECD guidance document on grouping of chemicals 194, which was last updated in 2014, at last is being updated for new edition that will be completed next year, and that's having its omics and molecular-based grouping guidance updated. So so that's important. And there's a new OECD Working Party for Hazard Assessment project came onto the work program earlier this year, and the kickoff meetings this month. And the project also runs starting now through end of 2023, and that's reporting specifically for omics-based grouping. So it's building on the work that has been done by Carol and Josh and the whole OORF team, and it's actually focusing specifically on grouping. So I must stop. I'm going to skip over. That was a little bit more on that reporting framework. That was to simply remind you of hopefully the journey that I'm 5 minutes over in terms of how long I was hoping. The journey I've taken you on through a little bit of justification around the metabolomics, leading through the case study, introducing the need to defragment this highly fragmented metabolic biomarker data to facilitate mechanistic interpretation. Small side tour out, just a few slides on, we really need to push this capability of metabolomics around the ADME-TK. And why are we interested in it? Because we'll bring that extra information into the grouping, because we're gaining information on the metabolism. And then a very rapid snapshot on some other highly important areas as we think about this type of NAM going forward into regulatory application. And I will not name names, I will simply show this. Thank all an amazing bunch of collaborators and colleagues on this slide, and say thank you for listening. Back to Carol or Connie.
Great. Yeah, I'll help moderate the questions. So if folks have questions, they can raise hand or put it into the chat, and we've gotten some in the chat already. So thank you so much for this talk, Mark. You're absolutely fine on time too, so no worries there. So there was a question from Leela early on that asked if your team has used this technique in the evaluation of ADC compounds, which is antibody drug conjugates. And maybe just a little broader, maybe you could talk about what kind of compounds you have tried in this model.
So the first question is easy because it's simply no. No antibody drug conjugate work at all. Well, I've been applying metabolomics. The first study was in 2002. So we don't want to go back through 20 years. There isn't any particular focus. More recently, in the last few years, it's been industrial chemicals more, but that's partly because of the excellent interactions and relationships that we've had with ECHA, and then obviously there's a steer there towards industrial chemical. So some of the, phthalates have been looked at, some work on and ongoing work around bisphenols. There's no particular focus on substances. Mm-hmm. And I don't want to waffle because I'm conscious of time and several questions.
No, that's great. So Carol had a question in the chat, but she also has her hand up. So I'm just going to unmute her and let her maybe go ahead and talk.
Mark answered my question, which was in the chat. I guess I sort of jumped in too early, Mark. I was thinking about how the MTOX 700 plus is similar to the S1500, but you got there. Really great talk. I love these case studies that are done in collaboration with the regulatory community. And I had a couple of questions about your case study. So a major issue. So first of all, I was wondering, you mentioned that ECHA was rejecting a lot of the arguments for the groupings. Yeah. And so there's two parts to my question. One is, did you change their mind? Did you provide them with the tool that would convince them, that they don't need anything further other than omics to help them with the grouping? Well, how did they feel about this at the end of your case study, your ECHA colleagues? And the other is associated with that in that, from my understanding, azo dyes are mutagenic carcinogens. And so I was curious because I can't think about how metabolomics would help you to identify that particular hazard, which would maybe have different considerations in a regulatory assessment than reprotox. But pairing it with the omics, with the transcriptomics, could really still identify that potential for mutagenicity. So I wondered if you could comment on whether you saw that. I didn't see it from the pathway analysis. But I don't really know about the pathway analysis in Daphnia either. So just those two and how those go together.
Sure. So case studies as a vehicle, I couldn't agree more with you. And multi-stakeholder case studies, I truly believe this is the way that NAMs are going to be progressing towards regulatory application. That wasn't a question. That didn't answer a question, but I'm just agreeing with something you said. Question one. Gosh, I don't want to be on record speaking for my colleagues at ECHA, as you will understand, because I have great respect for them, and it would be inappropriate for me to say they have said. Dun, dun, dun. However, I will say so. I'll say it in a different way. Their interest to come in and support and be part of the Cepheic matching omics grouping study is, I think, another indication of their interest in this. I hope it's okay for me to say, I don't know if any of them are actually on the call, that we have a new project with them that's actually started just about a week ago, which also includes a more detailed look into that third step in the omics-based grouping workflow. So that how much more information around the mode of action can we get? So I think it's fair to say that they remain very interested, and it is a categorical fact they have not said this won't work. So
fair. Yep. Great.
Yeah, yeah. And then in terms of the final question, you're right in a sense. Yeah, azo dyes were selected, and the classic, as you say, mutagenic effect in Daphnia, question mark, don't know. Mm-hmm. So mm-hmm. I can't answer the question directly. I don't know even if that particular mutagenic endpoint could occur in Daphnia. But what I will say is we have limited information on the on the metabolism of the dyes from the metabolomics data, and we've seen no evidence of the aromatic amines which need to form as part of the mutagenic pathway. So based upon the metabolomics and looking at metabolism of the dyes, there's no evidence, at least in a 48-hour study, that we could see the very substances which would be associated with the endpoint that you're discussing.
I see. Okay. So that might explain why you don't see, for example, P53 or something else at the top. I don't know much about P53 response in Daphnia, but
it could. Yeah, I'm providing a sort of a bit of peripheral information to say there's no evidence of this mutagenesis being important in Daphnia.
Okay. Yeah, it'd be interesting to try it in human cells and culture as well to see how the grouping would pair. And I guess with pairing of metabolomics and transcriptomics in vitro, the metabolomics would provide some of the information necessary for the high throughput toxicokinetics or whatever for the in vitro to in vivo extrapolation modeling. So you could actually do that reverse dosimetry piece using your metabolomics data.
Where we need to, we can demonstrate that the assay can measure the test substance. But something that we have to think very carefully about is what is the experimental design, the original experimental design. So if you want kinetics, you obviously need time course. Are you going to have a time course in a study that's looking at biological response? Well, ideally, yes, but sometimes cost doesn't allow it. And then also, what about the dose levels? If you're looking to obtain information on ideally mode of action, you're going to be perhaps at a higher dose, or concentration, sorry, if it's in vitro, you perhaps would be dosing at a higher concentration than if you want to get kinetic data and you actually want to look at clearance of parent, then you want to be dosing, but your concentration wants to be closer to km, Kmax. So we need to think carefully, if we're going to try and get TK and TD information out simultaneously, we actually have to go back to the design of the original exposure study and think carefully about how you actually conduct a study that's going to give you information on both of those. So it's not just the assay that needs to be capable of measuring both aspects. The design of the experiment needs to be fit for purpose to get information out that's on both aspects.
Excellent. Thanks so much, Mark.
Sure.
Yeah, thanks, Mark. I had a quick question too. We went through the example with Daphnia. We've talked a little bit about in vitro cells, human cells. Could you maybe comment on the species extrapolation of metabolomic pathways? Are certain ones well conserved? Is it unknown?
Okay, so my view on this has changed, because if you go back 15 years-ish, or 10 to 20 years, somewhere in that, I used to say one of the great advantages of metabolomics is if you're measuring lactic acid is lactic acid. It's the same in humans as it is in vitro, in vivo. It's the same in an earthworm. It's the same in a bumblebee. It's the same in Daphnia, and it is. So if your assay measures lactate, you can measure it easily in any biological test system, and that's an advantage. Well, it is, as long as lactate's conveying the same information in each of those biological test systems, and it isn't, for the very reason that you're highlighting, the conservation of pathways. So lactate, of course, is the end product of anaerobic glycolysis in us. In marine invertebrates, succinate is the end product of anaerobic glycolysis. So you've got to be very careful about the point that you're raising, the conservation. Now, in general, central metabolism is pretty highly conserved. So a lot of the pathways, a lot of the metabolic pathways will be conserved across species. But there was, the terminology in the metabolism world is primary metabolism and secondary metabolism. Loosely speaking, primary metabolism is the more conserved stuff, and the secondary metabolism is all the weird and wonderful molecules that, the cabbage makes and the strange moth makes, and the diversity of metabolites involved in secondary metabolism is vast, and it will be far less conserved across species. So I think I've seen some terminologies, and we've even used some terminologies, phylometabolomics, and equivalent to phylogenomics. So looking at the conservation of pathways, metabolic pathways, and there's certainly more work that could be conducted in that space as well.
Yeah, I mean, it's really important on the human health side is we use animals to go to humans, but there's a lot of work in the eco space. If, you test 3 species, but you want to protect all of them.
Yeah.
So I could see that being really important. So thanks for that answer.
Yeah. Well, and that I must just add John Coburn, who's my other half in Michabo Health Science. John Coburn leads the Precision Tox Consortium, the 20 million euro project in Europe, the Horizon 2020 project that a number of you, I'm absolutely sure, will be aware of. And the very heart of that project is to look at cross-species toxicity pathways across a series of models: Daphnia, Xenopus, C. elegans, Drosophila, also including human cell line. And there's over 10,000 samples will be analyzed for both transcriptomics and metabolomics in that study over the next. It's a 5-year project, so a lot of good work will come out on exactly that topic, Connie.
Great. Well, thank you very much, Mark. I wish we could talk longer, but we're at time. So thank you for that presentation, and we appreciate you coming to the meeting.
Thank you very much for the invitation.